My name is Helena Faust, and I'm going to give you a lecture about natural history of the human papillomaviruses. I'm going to talk to you about life cycle of human papillomaviruses, about immune responses to human papillomaviruses, and finally, I'm going to talk about HPV prophylactic vaccines. So first of all, let's keep in mind that viruses are parasites. What does that mean? That means that these viruses are not able to reproduce without host cell, and they have only minimal amount of own protein encoding genes, and the rest they will just hijack from the host cell. Viruses desire to look like an innocent part of the host cell, because otherwise they will be discovered as a foreign part and will be destroyed immediately by the immune system. Here are some facts about HPV. These viruses have skin and mucosal tropies. They are very host specific. That means that humans shouldn't be getting any viruses from animals. There are at least 200 human papillomavirus types already discovered, and that means they are very common viruses, and approximately more than 80% of the population is infected at least with one type during their lifetime. But good news is that usually this infection is asymptomatic and doesn't cause any disease. HPV is parasiting in host. It needs host cell for infection, reproduction, and spreading. So that's how looks one HPV. It has major protein L1 capsid here with green color. They have L2 with red color. And then they have their own genome here. Papillomavirus structure is called that it's icasohedral. It has diameter of 50 nanometer diameters. 5 L1 molecules consist of capsomeres. That means this one here is capsomere. If they gather together, it feels like kind of spheric, like a ball-like structure. Their genome is about 8,000 base pairs and encoding usually 8 genes. And every capsid has about one capsomere with 5 L1 molecules contains 1 L2 molecule. So how does the virus get into the cell? First of all, we have to know that this virus has to actually end up in nucleus, because there the replication and transcription takes place. The virus binds to the receptor, but so far unknown, and will be trafficked in endosomes. The process is called endocytosis. In late endosomes, what happens there is that L1 capsid is degraded, but L2 binded with genome complex, this moves to the nucleus, and there where starts transcription of some HPV genes and replication. HPV genome, it contains only minimal amount of needed genes. It's 8 open reading frames and regulatory region. There are early genes and late genes. Early genes are E1, what stands for replication, E2, what stands for replication and transcription, E4 for viral release, E5 for immune evasion, E6 what binds P53, and E7 which binds with BRB. E6 and E7 are known as oncogenes because they are making cells to divide, meaning they are modulating the cell cycle and making cells to divide where they shouldn't do it. You can also pay attention that E1 gene is large. That means that encodes some larger protein. Same with the late genes. L1, what's a major capsid protein, and L2, which are minor capsid protein, are also large, about 500 base pairs. So L1, L2, and E1 takes a majority of the base pairs of the genome. HPV life is tightly regulated along with differentiation of the cells. Down here, our basal stem cells, these are allowed to divide, and these cells do wander upwards during their development to differentiate, and finally they will die and will be shedded off from our skin. For infection of the virus, it's needed a wound, or can happen also in transformation zone in cervix, where those kind of basal cells basically naked and very vulnerable. At first, in lower epithelial, HPV is very silent. The replication is just to maintain genome, but more further, the cell develops more intense, starts replication, and also expression of the HPV proteins, what's needed. For example, E6 and E7, which makes cells to divide. And also replication proteins, L1 and L2 are needed for E1 and E2, which are needed to genome amplification, will be intensified here in the upper layers. The uppermost layer will be E4 expression and capsid protein L2 and L1 expression. Here, in the naturally dying cells, the virus particles will be released. We have to keep in mind that cells are manipulated by HPV. So, what's the most important here is E6 and E7, which binds to P53 and BRB, meaning that they're binding and degrading it and making cells to divide at a place where shouldn't do it. There are some difference in low-risk HPV infectious and high-risk HPV infection here. While in low-risk infection, this dividing of the cell starts a little bit upper epithelial layers. In high-risk HPV infection, it happens both in upper and lower layer. This slide shows with what E6 and E7 bind and what they do. Most important here is that E6 binds P53 and inhibits apoptosis. Same time, E7 binding BRB and degrading it. So now our cells are cycling and all the needed enzymes are there. What's needed for replication, like for example, DNA polymerase. And what is then needed from the HPV side? The needed proteins are E1 and E2. E1, it's a helicase, hexameric helicase, and it's a only HPV enzyme. How does that work? That works like that here we have double-stranded DNA, and it unwinds it so that after it will be single-stranded. And if it's a single-stranded DNA, then coming DNA polymerase to synthesize a new chain. E2 brings E1 to viral DNA, connecting viral genome to the host chromosomes. Also, E2 regulates E6 and E7 expression. And we have to keep in mind that the rest of the needed replication enzymes are stolen from the host cells. Later in life starts E4 expression, just before capsid proteins will be expressed. And this is a marker for active virus, and it's expressed in extremely high levels, so that 30% of the cell content can actually be E4 protein. And it's also forming koilocytes. Koilocytes are here. It's E4 expression is a marker for virus genome amplification success, virus synthesis, release, and transmission. It's shown that E4 can assemble into multimers and suppress cell proliferation and reorganizing keratin network so that the cells getting more fragile. E5 protein is a transmembrane protein, and it's forming ion channels, has transforming abilities, at least in some species. And what's the function for it is mainly that it triggers immune evasion by downregulating of cell surface proteins involved in the antigen presenting. Also, it inhibits apoptosis. For some HPV, E5 is needed also for genome amplification and switch to L1 production. Final stage is that virus genomes has to be packed and released. It starts with L2 expression. E2 brings L2 to replication regions, and L2 genome complex forms. These L2 and genome complexes will be packed into the L1 capsids. Such kind of virions will be maturing in dying keratinocytes and released in naturally dying cells. As it's in oxidizing environment, this supports disulfide bonds between L1 proteins. The result is extremely stable virions. Conclusions for that part. HPV gets into cell if it has access to basal membrane, for example, through microwound or access to basal cells. Receptor takes HPV into cells, and the virus could be trafficked to nucleus. HPV protein expression is strongly regulated according to the development of the host epithelial cells. E1 and E2 plus host own replication machinery is needed for HPV replication. E6 and E7 promote cells to divide and not to die. E4 is needed for viral release, E5 for immune evasion. L2 in complex with genome will be packed into L1 capsids in uppermost keratinocytes. Now we are moving further with the immune responses to HPV infection, because we have immune system, so that means it should react to the HPV and eliminate it as fast as possible. For your refreshing your memory, immune system is divided into the innate immunity, what's not so specific, but it's fast, for example, inflammation, and to adaptive. Adaptive is very specific, but it takes time. Adaptive immune system is divided into the cellular immunity, meaning T cells, and humoral immunity, meaning B cells, which produce antibodies. So how does it look like usually HPV infection? It starts very silently. We don't know when it actually happens. For a while, we have no signs about. But then may come some first lesion, for example, wart. And then DNA of the virus is also detectable. It might take weeks or months, and spontaneously the wart is regressing. That means that this was a sign of cell-mediated immune response. And then, after 1 year or so, most of the people have no DNA anymore detectable, while about 10 to 20% of people still suffer from the lesions and still have DNA detectable. Most of the people seroconverse to the major capsid protein L1 during this time, but not all. But it's shown that more persistent and higher copy numbers infection is, more likely the L1 seroconversion happens. So how should look like one successful immune response to the HPV infection? Well, here we have basically shown innate immunity, and here should come adaptive immunity from that side. So this kind of reaction should start with keratinocytes down here, which have pathogen recognizing receptors like toll-like receptors. These toll-like receptors, this should activate non-specific innate immunity response with pro-inflammatory cytokines like interferons. Those star-like cells here, they are antigen-presenting cells. They should pick up these cytokines, and they should travel to secondary lymphoid organ, for example, our lymph nodes, and spread that information. They should give it to the CD4 T helpers and memory CD8 cytotoxic cells. These cells should go back here and kill the infected cells. And T cells should give the information further to the B cells to remember that information and produce antibodies, that this infection would never happen again. So, but why do we then get such kind of long infection and sometimes even no seroconversionary signs of clearance of the infection? That's because HPV is very successful to suppress our immune system. First of all, very low levels of proteins are expressed, no viremia, no cell death, no lytic phase, no inflammation, global downregulation. HPV proteins E6 and E7, they downregulate interferon response, toll-like receptors, and also E2 regulates some receptors. Langerhans cells, which are antigen-presenting cells, their maturation is blocked by L2, and migration is inhibited by E7. So as there is no inflammation, keratinocytes do not release pro-inflammatory cytokines. There is no activation of Langerhans cells and dendritic cells. There is no stimulation for dendritic cells to activate, migrate to lymph nodes and process antigen and present it. Therefore, HPV evades innate immune response, and as innate immune response is delayed, the activation of adaptive immunity do not happen. So this is very, very successful strategy here for this virus. Conclusions for the immunology part. HPV avoids innate immunity through not being recognized. It lives silently in differentiating cells. Virus release happens in dying cells where immune system do not reach. Also, it blocks innate immunity activation cascade through E6 and E7. There is no pro-inflammatory signals like interferons. Also, HPV do not let antigen-presenting cells to do their job and create virus-tolerant environment. As innate immune response is not adequate, that in time, adaptive immune system do not react in time to come and eliminate it. this kind of HPV infection can persist long time without any disturbance, as E6 and E7 are carcinogenic and tell cells to divide continuously, and integration in the host genome happens very often, what causes also often loss of E2 gene, what regulates E6 and E7 expression, and therefore often E6 and E7 expression raises after integration event. Then it's already too late after integration event for immune system to do anything. This is a fatal damage and will cause cancer development. So how can we avoid HPV? So we are lucky there are prophylactic vaccines available. At the moment, there are 3 licensed prophylactic vaccines available. GlaxoSmithKline producing Cervarix, and Merck Gardasil 4, and next generation Gardasil 9. All of those vaccines are made out of virus-like particles and adjuvant, and all of them do contain virus-like particles of HPV 16 and 18, which are the most dangerous HPV types. Gardasil also gives protection against the genital warts, and Gardasil 9 rest of the oncogenic HPVs. So what's the difference between Cervarix and Gardasil? Basically, they have a different protein expression system. In Cervarix, it's insect cell line, and in Gardasil, it's yeast, and they have different adjuvant. So what is VLP, virus-like particle? It's consisting only with L1 capsids, which are spontaneously gathered together. They made such kind of spheric structures. In real viruses, there are also L2 and genome, but vaccines do not contain any other viral protein than L1. So here are the HPV types which are used in vaccines. 16 and 18, they are part of all of the vaccines. And they should prevent 70% of the cervical cancers. Gardasil contains also 6 and 11, what should prevent 90% of genital warts. And the rest of the non-valent vaccine types 31, 33, 45, 52, and 58, they should prevent about 20% of the cervical cancers. So how do the vaccines protect, actually? First of all, virus-like particles in vaccine elicit neutralizing antibodies, which then gonna circulate in bloodstream and tissues. What do those neutralizing antibodies do? They bind to the real virus when it appears and do not let it to enter through endocytosis, or they prevent uncoating from some endosomes. Also, it might be that it just promotes aggregation of the virus. And in the end, it's called that the virus is neutralized by neutralizing antibodies. So how is it so successful, those neutralizing antibody creation in human beings? That's because it's intramuscular injection directly to our bloodstream. That means that those virus-like particles end up very fast in lymph nodes and being taken up there. And this is a guarantee for the high level of antibody production. So if in natural infection, there has to be this cascade from innate immunity to the adaptive immunity, then this part is totally skipped in vaccination process. You also have to keep in mind that those L1 VLPs are very antigenic even without adjuvant. So what about antibody levels after vaccination? Here we can see that here are the natural infection levels, and these are the levels after vaccination. And it's like 100 times higher than natural infection. The levels after vaccination, they will stabilize and go time in time, but it has been shown that after 5 years, antibodies are still there. So, and if you give a booster dose, what's given here, antibody level raises back to the original or even higher stage. And this is a sign for immune memory. And high antibody levels, they are correlated with protection. So what about dosage? Originally were always used 3 doses, but now have been shown that if you use 6 months apart, 2 doses, it gives almost as good high level. And if you give just a 1 dose, it's still about 10 times higher than the natural levels. And that shows that even 1 dose gives protection. There is also a phenomenon called cross protection discovered during the vaccination. It's about that vaccine protects also against HPV 31, 33, and 45, which are close relatives to vaccine type 16 and 18, even those viruses are not included to the vaccine. Cervarix inducing more and higher level cross-protective antibodies than Gardasil 4 does, because of the adjuvant and conformation of VLPs are better in Cervarix. Cross-protection effect is explained by high levels of antibodies and by cross-neutralizing epitopes shared between close relatives. Conclusions for the prophylactic HPV vaccines are here. Those prophylactic vaccines, they induce 2 to 4 logarithmic higher levels of neutralizing antibodies than natural infection in blood and the mucosa does. Also, the avidity is higher in vaccinated subjects than subjects with having natural infection. Neutralizing antibodies bind to real virus and neutralize it, meaning they do not let it to infect the cell. It's proven that there is immune memory for L1 VLPs, B memory cells to remember them, and it's also some cross-protection due to cross-neutralizing epitopes shared between close relatives and high levels of antibody. So far, it has been shown that immune memory lasts at least 9 years. Thank you very much for listening me.